Welcome to biology, the scientific study of life. Transport in vascular plants. Notice this figure presents corridors. An overview of transport in vascular plants. Notice that. Let's start with roots. Roots absorb water and dissolved minerals from the soil. Notice that. The pathways, water and minerals are transported upward from roots to shoots as xylem sap. Notice that. 3. Transpiration, the loss of water from leaves mostly through stomata, creates a force within leaves that pulls xylem sap upward. 4. Notice that through stomata leaves take in CO2 and expel CO2. The CO2 provides carbon for photosynthesis. Some O2 produced by photosynthesis is used in cellular respiration. Notice that. 4. 5. Sugars are produced by photosynthesis in the leaves. Notice also returns or sugars are transported as phloem sap to roots and other parts of the plant. And notice that roots exchange gases with air with the air spaces of soil, taking in O2 and discharging CO2 in cellular respiration. O2 supports the breakdown of sugars. Proton pumps provide energy for solute transported. Notice that cytoplasm, ATP, H positive, and extracellular fluid, H positive. So proton pump generates membrane potential and gradients. Notice that proton pumps provide energy for solute transport, pumping H positive out the cell. Proton pumps produce an H positive gradients and a charge separation called a membrane potential. These two forms of potential energy can be used to drive the transport of solutes. Solute transport in plant cells. Notice that membrane potential and cation uptake. Cytoplasm, extracellular cations K positive, for example, are driven into the cell by the membrane potential transport protein. Notice that co-transport of anion. Cell accumulates anions in O3-, for example, by coupling their transport to the inward diffusion of H positive through a co-transport. Notice that co-transport of natural solutes. Plant cells can also accumulate neutral solutes such as sucrose by co-transporting down the steep proton gradients. Water potential and water movement, an artificial model. Notice this is abbreviated letter by the Greek letters Psi. Psi, this is as translated or abbreviated called water potential. Water potential and water movement, an artificial model. In this Y-shaped apparatus, a selectively permeable membrane separates pure water from 0,1 solution containing a particular solute that cannot pass freely across the membrane. Water moves across a selectively permeable membrane from where water potential is higher to where it is lower. The water potential Psi of pure water at atmospheric pressure is 0 MPa. MPa, which mean megapascals. If we know that the values of pressure potential Pcp and solute potential Ps, we can S, which means fluxed and solute potential, we can calculate water potential like this: psi equal psi p plus psi s. The values of psi and psi s in the left and right arms of the tube are given for initial conditions before any yet movement of water A. The addition of solutes reduces water potential to a negative value. B plus C, application of physical pressure increases water potential. D, a negative pressure tension decreases water potential. Notice that water relationships in plant cell. In this experiment, identical cells initially flexed are placed in two different environments. The photoblasts of flexed cells are in contact with their walls but lack turgor pressure. The blue arrows indicate the initial direction of net water movement. Notice that initial flexed, psi p equals 0, psi s equal minus 0.7, psi equal minus 0 s MPa, which means megapascal. Plasmolysed cell an osmotic equilibrium with its surroundings. Psi p equals 0, psi s equal minus 0.9, psi which equal 0 minus 0 p megapascal. Initial conditions: cellular psi more than environmental psi. The cell loses water and plasmolyzes. After plasmolysis is complete, the water potentials of the cell and its surroundings are the same. Notice the distilled water triggered. Notice the initial conditions: cellular psi less than environment psi. There is a net uptake of water by osmosis, causing the cell to become turgid. When this tendency for water to enter is offset by the C or the back pressure of the elastic wall, water potentials are equal for the cell and its surroundings. The volume change in the cell is exaggerated in this diagram. Notice that a watered notice the plant regains its turgor. Notice that cell compartments and roots for short distance transport. Notice that cell walls, cytosol, vacuole transport proteins in vascular membrane, regulate traffic of molecules between the cytosol and the vacuole. Also, the transport proteins in the plasma membrane regulate traffic of molecules between the cytosol and the cell wall. Notice cell compartments. The cell wall, cytosol, and vacuole are the three main compartments of most mature plant cells. Notice that symplastic root and transmembrane root. The symplast or symplast is the continuum of cytosol connected by plasmodesmata. Notice also the epoplast is the continuum of cell walls and extracellular spaces. Epoplastic root. No transport routes between cells at the tissue level. There are three passages: the transmembrane, symplastic, and epoplastic root. Substances may transfer from root to another. These are represents cell compartment and routes for short distance transport. Notice also the lateral transport of minerals and water in roots. Uptake. Notice the epoplastic root. Uptake of soil solution by the hydrophilic walls of root hairs provides access to the epoplast. Water and minerals can then soak into the cortex along the matrix of walls. 2. Minerals and water that cross the plasma membranes of root hairs enter the symplast. Notice 3. and soil solution moves along the epiblast. Some water and minerals are transported into the protoblasts of cells of the epidermis and cortex and then move inward via of symplast 4. Notice that within the transverse and radial walls of each endodermal cell is the Casparian strip, built of waxy material that blocks the passage of water and dissolve minerals. Only minerals already in the symplast or entering that pathway by crossing the plasma membrane of an endodermal cell can detour around the Casparian strip and pass into the vascular cylinder. Notice 5 also endodermal cells and also parenchyma cells within the vascular cylinder discharge water and minerals into their wall epiblast. The xylem vessels transport the water and minerals upward into the shoot system. Notice also the closer look pathway along epiblast pathway through blasts and Casparian strip and endodermal cell. Notice that. More about mycorrhiza, symbiotic associations of fungi and roots. The white mycelium of the fungus ensheaths these roots of pine trees of fungal hyphae provide an extensive surface area for the absorption of water and minerals. Guttation, root pressure of forcing excess water from this strawberry leaf. And notice also the generation of transpiration pull in leaf. The negative pressure at the air-water interface in the leaf is the physical basis of transpirational pull, which draws water out of the xylem. Notice when in transpiration water vapor diffuses from the moist air spaces of the leaf to the drier air outside via stomata. Notice too also that at first the water vapor lost by transpiration is replaced by evaporation from the water film that causes mesophil cells. Notice 3 step, or notice here, evaporation causes the air-water interface to retreat farther into the cell wall and become more curved. As the rate of transpiration increases, as the interface becomes more curved, the water film's pressure becomes more negative. This negative pressure of tension pulls water from the xylem where the pressure is greater. Notice the Epsi equal 10,000 pm megapascal. Xylem sap ascent by bulk flow. Ascent of xylem sap hydrogen forms an unbroken chain of water molecules extending from leaves all the way to the soil. The force that drives the ascent of xylem sap is gradients of water potential passy for the bulk flow over long distance. The passy gradients is due mainly to gradient of the pressure potential passy P. Transpiration results in the passy P at the leaf and of the xylem being lower than passy B. At the root end, the passy B follows. Values shown at the left are snapshot during daylight. These specific values may vary, but the direction of the water potential gradients remains the same. Notice that transpiration, cohesion and addition in the xylem, water uptake from soil. Notice the water potential gradients. Xylem, sap, mesophil cells, stoma, water molecule, atmosphere, xylem cells, addition cell walls, and closer look. Notice also water molecule, root hair, soil particle, water. The last one or the last notice: open stomata left and closed stomata. The differences of stomata help regulate the rate of transpiration. Effects of transpiration on wilting and leaf temperature. Change in garcelle shape and stomatal opening and closing surface view. Notice also garcelles of typical angiosperm are illustrated in their turgid stoma open and flexed stoma closed states. The pair of garcelles buckled outward when turgid. Cellulose microfibrils involve resistant stretching and compression in the direction parallel to the microfibrils. Thus, the radial orientation of microfibrils increases the two garcelles are attached to at their tips, so the increase in length causes buckling. B. Role of potassium in stomatal opening and closing. Port of K positive potassium ion symbolized here as red dots across the plasma membrane and vascular vacuole membrane causes the turgor changes of garcelles. The mechanism of stomatal, these are represents the mechanism of stomatal opening and closing. Notice also the structure adaptations of leaf. Notice the